started. I want to thank everyone for joining us for this first experiment at doing a journal club through Facebook Live. My name's Tony Blau. I have been a professor and a hematologist and a scientist at the University of Washington in Seattle for more than 30 years. About 5 years ago, I developed multiple myeloma, and as a result of developing myeloma myself, I came to appreciate a problem that I hadn't understood in the same way previously, and that is, as a patient with myeloma, I've had the good fortune of having many good treatment options, but the downside of having many options is that it's really difficult to know which option was best for me at a given point in time. And so what I've most wanted to know is what were the treatment choices and outcomes of every other myeloma patient. And because that information didn't exist, I started All4Cure. And so we have a wonderful team here in Seattle, and we are trying to figure out different ways of being of service to the myeloma community. Our main effort is through our platform, our online platform, but we have launched a number of other initiatives through Facebook to provide educational material, kind of fun videos that describe basic concepts relevant to myeloma, and then a deeper dive into the personal stories of some of the participants in All4Cure that's been, very interesting and rewarding for me to read, to get to know people more as individuals. And so the idea for what this next hour is about, it comes from Dana Holmes. And Dana will shoot me a Facebook message every now and then. And Dana said, could you help distill this paper that I'm showing here into kind of comprehensible language that would be accessible to lay people. And this reminded me very much of an activity that happens very frequently at academic centers throughout the country, universities throughout the country, where there is this notion of something called a journal club. And so a journal club will typically meet once a week. Faculty and trainees will come together, and someone will present a paper that is of interest and distill it down into the core nuggets that make it easy for now a whole room full of people to try to understand the implications of the paper and its significance to their interests and their understanding of science. And so this is an experiment, like I say, of trying to extend the journal club concept now to the Facebook community. And so this is an experiment in part because we are neophytes in using Facebook Live and in trying to understand how to adapt what I'd like to convey to this platform. So please be generous in forgiving any sort of technical problems that we might have along the way. And I'll just go ahead and dive into how I think of this paper that was published. It's actually now published this coming month in October of 2020 in a journal called Nature Reviews Cancer that is describing comprehensively this notion of cancer driver genes. And so, again, welcome. There is a lag in the communication that happens on Facebook, and to facilitate our interactions, I think it will be most helpful if we save the questions to a time where I can see them. I have other members of our team standing by to try to help make sure that I hear and respond to questions as they come up. And so what I thought might be interesting to try, and we'll see how this works, is to provide kind of very much in the same way that I would present to a bunch of professors in a room a paper, kind of having a whiteboard so I could show you how I think about this stuff. And so I think there are some important background things to kind of keep in mind that I think are useful in trying to understand the context of this paper. And so the first concept that I think is important to have some idea about is the concept of a cell. And what does a cell look like? And in our bodies are made up of lots of cells. And in general, there are really two parts of the cell that are important to remember. There's the external membrane that separates the cell from the rest of the world. And then there's this inside the cell part called the nucleus. And this is where DNA is housed. And so I'll talk about that a fair amount. Stepping back for a moment, it's important to know that, and you guys probably already know, that DNA is really the blueprint for our cells, every cell in our body. My DNA is unique to me. It's different from anyone else in the world. And those differences in DNA are the reason that I'm built differently from anyone else. And so you can think of DNA as this blueprint. And it would be useful to know a little bit about how this is organized. And so the way I think about it is DNA is built on fundamental building blocks called nucleotides. And there are really four kinds of nucleotides to keep in mind. And they just go by letters, so it's pretty simple. There's A, G, C, T. So there are four different kinds of building blocks. And the way that they're organized is that they're organized into these long chains that are similar to the chains in a chain link fence. So if I were to link these chains together, you could say this is A, C, T, C, A. I just made that up. But that's different, and the cell recognizes that differently than if, for example, you had 5 links that had a different order. So, for example, G, C, C, T, A. Totally different information. Depending on the nucleotides and their sequence within this chain of nucleotides that comprise DNA. And so so that's one important principle. The second thing is, how many different nucleotides, individual building blocks, are there within the nucleus of a cell? And for humans, we have about 6 billion of these individual nucleotides lined up, linked up in very specific sequences that are unique to us. And so, that's kind of an amazing number. 6 billion of these things that make us us. And one question you might think about is, every cell in our body, pretty much, and I'm going to generalize a little bit in some of my discussion, in some of my comments. So if you're a real aficionado on this stuff, you won't have too much difficulty in finding, disputing something or another that I say in the next hour. But pretty much, all of the cells in our body have the same DNA sequence, the same 6 billion nucleotides arranged in the same way in every cell. So, and that's unique to me. If we looked at somebody else's DNA, that would be a sequence that is unique in every cell of the body of that other person. Okay? So you could say, well, heck, if every cell in our body has the same sequence of same genetic blueprint, the identical genetic blueprint, then why doesn't every cell in our body look exactly the same? Why aren't we just blobs of cells where every cell is looking exactly the same because they all have the same genetic blueprint? And so it seems like a little bit of a contradiction there, and I'll explain that. So let's think about it for a moment. Let me get rid of this. And think for a moment of, let's think about the cells in our body and all of the different kinds of cells that we have in our body. So, one question that I think is important as I think about a number of things is, how many different cells are there in our bodies? How many different cells does each of us have in our bodies? And the real answer to that question is nobody really knows the answer to that for sure. But there are some pretty good estimates that each of us in our bodies have about 37 trillion cells. And that includes brain cells, heart cells, there are different kinds of brain cells, fat cells, muscle cells, yada, yada, yada. So we have, an enormous number of cells in our body that all share the same genetic sequence. And so why do some cells that have the same genetic blueprint make brain, while another cell of our body with the same genetic blueprint makes muscle? And the reason for that is that each of us have the same DNA sequence, and you can just, and these long chains of nucleotides make up these bits of DNA called chromosomes. And so each chromosome will have hundreds of millions of these nucleotides. We have 46 chromosomes in total. Men have XY. But the bottom line is that we have the same genetic blueprint, but there is a whole other layer of control within a cell that specifies what part of the DNA is active in a cell for a given cell type. And so I've already kind of explained this idea of nucleotides, and there being 6 billion of those. But if you look kind of, and this is totally conceptual, if you look at a long stretch of DNA, let's just imagine it's a long stretch of DNA. I've taken a piece of a chromosome, stretched it out. What you find is that there are regions in the DNA that I'll show here as boxes that form areas called genes. And these genes are important because they make protein. And much of what we see in a cell that allows us to see whether it's a muscle cell that has muscle proteins versus a brain cell that has a whole different set of proteins, myelin and things like that, has to do with the genes that are turned on or off in that given cell type. So even though every cell in our body has the same number of genes, and it's estimated that we have about 20,000 genes in our, and it might be slightly lower than that, I think, in the most recent estimate, different cell types will activate different genes within that cell, and they'll silence other genes within that cell. So even though we all have the same genetic blueprint, there's this extra layer of control called the epigenome that involves things like chromatin and DNA methylation and things like that determines which genes are turned on or off in a given cell type. And that's why we're not just blobs of all the same cells. Each cell has the same genetic blueprint, but it also has this extra layer of control that determines which proteins are made in a given cell based on the activity of the genes that are responsible for making those proteins. Okay, so that's a little bit more background information. So, I guess let me just, I know there's a little bit of a lag, but is there, and this might be, I just wonder, are there any questions? Let me just see, are there any questions that have come up? I'm just looking to my team. No, things seem okay. So let me continue then. Yeah.
Bonnie just asked a question.
Yeah.
Does each type of cell have a stem cell?
Oh, Bonnie, that's a good question. Does each type of cell have a stem cell? So there are lots of different cell types in the body, and lots of different tissues, and there are many tissues for which people have identified stem cells. And so you guys will know that, bone marrow is a tissue, in the hollow spaces of our bone. It's a blood production factory. And obviously in the bone marrow there are a small number of stem cells that expand, massively to make all the different types of cells in the blood. So bone marrow definitely has stem cells. But there are also, intestinal cells definitely have stem cells. That's super clear. There are, there are some stem cells in brain. I think some of the cells in the brain can be renewed from a stem cell population, but this is, I think, fairly limited. And in a tissue like heart, my colleague Chuck Murray, who's a world expert in this area, spent 20 years trying to find heart stem cells in adult hearts. And despite a lot of controversy by a lot of researchers around the world, the heart, for example, does not appear to have an adult population of stem cells that can regenerate heart. So, muscle, skeletal muscle also has stem cells. So there are a number of different cell types that do have stem cells in adults, but certainly not all. Not all adult tissues appear to have stem cells. So, any other questions? Jane? Oh, we're okay? Okay, so let me continue. So in this paper, so if, I have myeloma, I think a lot about myeloma. I'm sure many of the other people here think about multiple myeloma. And so what in the heck happened to us? How did we get this thing? And what was the process that led to this? And so you could think, so you can think about, multiple myeloma comes from a type of cell called a plasma cell. And so plasma cells make antibodies. They have very important functions in our immune system. And so they're, really key, super important cells. And multiple myeloma comes from, I am guessing, I don't know the answer to this for sure, but I'm guessing that multiple myeloma probably doesn't come directly from a plasma cell, a mature antibody producing plasma cell. Multiple myeloma probably comes from a precursor cell that would, under normal circumstances, have been destined to make a plasma cell. But now, this cell has acquired a mutation that allows it to live longer than it would normally. And this mutation, this is part of the topic that's brought in this Cancer Review article, is what are the kinds of genetic changes that can lead to the development of cancer and can forward, cancer progression. Because even though I've told you there are 20,000 genes, not all of them have equal contributions to forming or propagating cancer. There are a subset of these genes, and it's probably a little bit more than, 500, I think it's 560 or so of these genes that have been heavily implicated in the formation of cancer or in sustaining cancer. And these are called cancer driver genes. So let's look for a moment. Let's go back to, this scenario where we have this cell that was destined to make a plasma cell. And it is every time a cell divides, or when a cell gets exposed to some toxin, something wrong, or even under normal circumstances, just under normal cell division, there will be some small number of mutations that happen with each cell division. And so let's imagine this is a precursor cell and it makes two daughter cells. Those two daughter cells, if you look super carefully, will be very slightly different from the mother cell from which they were derived. And this is one of those areas where I'm probably on a little bit of thin ice, but the way I think about it is with each cell division, you probably have somewhere in the order of roughly six errors. And that means out of 6 billion bases, maybe 1 in a billion, ends up being different from the mother cell. And that's just with normal cell division. So if you think across 6 billion bases, and you just randomly, have errors in a tiny fraction of those, let's just look at this long thing of DNA again, and I show you these genes. These areas of the DNA that contain genes is a very tiny fraction of all of the DNA in the cell. These gene encoding parts of the DNA are only about 1% of all of the DNA. There are vast stretches of this intervening DNA that has important functions that are getting figured out more and more, but there's still a lot of unknown aspects of what this non-coding DNA does. But if you're just going to say, okay, I'm going to take, now this is just like I said, this is just 1%. These parts of the DNA that have genes are only 1% of the whole genome. So if you say, okay, out of 6 billion bases, I'm going to say, okay, I have a mutation here, I have a mutation here, I have a mutation here. The vast majority of mutations that happen within a cell have no functional consequence. You could have, cancers often have thousands, sometimes tens of thousands of mutations, but the vast majority of those mutations have no bad consequence. So you could say, this cell has acquired, this cell's acquired, some mutations. If those mutations have, no functional consequence on how the cell behaves, then who cares? There are mutations that could happen that could be deleterious to the cell. Okay, this gene that happened to get mutated was really important to that cell's survival. And so this cell, let's say, unlucky cell that it was, it acquired a mutation that it caused it to die. And now it doesn't get to be, live anymore. But again, from the cancer point of view, who cares? I don't care about a cell that's acquired a mutation that causes it to die. I care about the tiny, minuscule fraction of mutations that happen to land in a region of the genome that actually make the cell live a little longer. And so this cell acquired, some mutations, and just by total chance, one of those mutations might have landed in an area that allows that cell, let's say that cell was supposed to live for 4 months, but now with this mutation, it gets to live for a year, 12 months. Now, if it comes, if it lives for a year and it dies in 12 months, who cares? But let's imagine a scenario now where this cell lives longer and it has the opportunity to acquire additional mutations. And so let's say right at month 5, it gets another one. Maybe that allows it to live even longer, and then it gets another mutation down the road here. And it's this sequential buildup of mutations now that allows a cell to go from being a single cell to now when this cell divides, let's say it's this naughty cell that's thinking of doing nefarious things, now when it divides, each of its daughters have all of the mutations that were present in the mother cell, and they again have the possibility of now acquiring additional mutations. And so it's like a tree where this is a trunk, but then the branches of the tree, each cell that comes from that tree has its own trajectory, if you could say mutational trajectory, where now you have this dominant, cell type where it has, let's say, mutation 1, 2, 3, and now it's grown. But now its daughter has acquired mutation 4, 5, 6, and this has acquired mutation 7, 8, 9. They all come from the daughter, but now things are getting complicated because you can imagine that these cells, some of these mutations are going to make these cells behave differently. Some of these mutations might make these cells resistant. You could imagine this clone of cells could be resistant to drugs that work great to kill some of these earlier cell types. And so cancer, this is kind of a general issue for cancer, generally is this is an important point that I think people don't understand, appreciate maybe adequately sometime. But in a person with cancer, every cell in our cancer is slightly different from every other cell. And that sounds super scary. I'm not meaning for it to sound scary, but you can imagine these are my cancers. Let's imagine I have, 5 cancer cells in my body. They will be, each one will be very, at least very slightly different from every other cell. And so as we're trying to think about ways to keep our cancer under control, we're not just trying to think about how to, it's not a monolithic, homogeneous population of cells. They're kind of an unruly group of cells that you're trying to corral and bring down to the lowest level possible. And so now when you think about this whole concept of, now let's finally get to discussing a little bit about this paper. And I'm just going to quickly just double check the time here to make sure that am I okay? Oh yeah, I'm okay on time, it looks like. So as you're looking at the concept of cancer driver genes, there are, our cells in patients with cancer have lots of different mutations. There are lots of different classes of mutations, types of mutations that can happen in a person with cancer. And this paper focuses on mutations that are called point mutations.
Hey Tony, before moving on, we have a couple of questions from a couple of people.
Yeah, thanks, Shane.
We have a question from Andrea asking, how do infections like Epstein-Barr impact cell mutations?
Mm-hmm. Okay.
And after that, Bonnie says, at some point, would you please connect the dots between individual myeloma cells carrying certain mutations and clones and subclones?
Yes, let me try to first address the Epstein-Barr virus question. So Epstein-Barr virus is a virus that I think is episomal, so it causes B cells, a certain type of cell, a B cell, which is one of plasma cells or a type of B cell, can be driven to replicate to very large numbers. And there are malignancies that can be associated with that. So most people with Epstein-Barr virus, they have a proliferation of B cells. The virus gets under control and they get better. But there are malignancies associated with Epstein-Barr virus where the proliferation of B cells is accompanied by mutations that lead to a B cell malignancy. And so that's probably just about all I know about that. I don't know a whole lot more than that, other than there are clearly malignancies associated with, not very common malignancies associated with Epstein-Barr virus. Trying to get at Bonnie's question, I'm not sure I'm understanding all of it. You know, the idea of a clone is a cell population that comes from a single cell. So this is one cell. It divides and it makes, you know, this whole population of cells. Now, and you would call that a clone. Now, what I have already said is that every time a cell divides, there will be subtle differences from one cell to the next. And so, in fact, these cells won't be identical to each other, ever. So the concept of a subclone really pertains to a subpopulation of cells where the genetic change that has occurred has some functional significance. So let's imagine that this subclone, you know, these, let's say this, all of these have, you know, mutation 1, 2, 3, and kind of in a, I think in a probably easier to understand way, you could imagine in colon cancer, for example, you could have a mutation in a gene called APC. And now, you know, all of these progeny cells have a mutation in APC. But let's imagine here now, these cells acquire a second mutation of an important oncogene called KRAS. And so, and that has a lot of functional significance. So the term subclone will typically refer to a subpopulation of cells arising from the original clone that have acquired a second mutation, two or more mutations that have a functional significance that make them take off. So in this case, for example, this would be the typical prelude from going from a colon polyp to colon cancer. When that subclone now takes off, has two mutations, and causes, you know, has a functional impact. So it's a little bit of a mushy definition, the idea of a subclone, but it's typically used to refer to cells that have acquired a second level of mutations that have some functional impact. So, so in thinking about the kinds of mutations that, you know, are described in this paper, as I said, this mainly describes point mutations, which in a point mutation refers to this scenario. Remember this little chain link fence. This is A, C, G, T, C, A. And now this has changed to 4, 5, 6. This has changed to A, G, G, T, C, A. So this C has changed to this G. That's a point mutation. You could say, heck, it's only 1 out of 6 billion frickin' bases. How bad can that be? But it depends on where that lands. It can be pretty darn significant depending where that change occurs. This is also called a single, an SNV, or single nucleotide variant. And let me just step back for a moment and make a distinction between germline DNA and somatic DNA. Germline DNA is what I was referring to at the beginning of this session, where each of us are comprised of a unique sequence of 6 billion bases that's unique to us. Makes us different from anyone else. And we pass that along to our progeny. We pass off along half of the complement of our DNA to our kids. And so germline means this is what's passing through generations from one person to, one generation to the next. Somatic DNA is what you think about when you're thinking about mutations that are associated with cancer. And so, for example, when I have, let's get back to the plasma cell example that I was talking about. Here's a plasma cell. This is the precursor cell that can make a plasma cell, or it can acquire mutations that cause it to make multiple myeloma. These mutations are not present in my germline DNA. They're not present in any of the other cells of my body. I'm not going to pass along these mutations to future generations. These are mutations that happen only in my tumor. Now, there are somatic DNA changes that happen in other cells of our bodies over time. We're constantly acquiring these somatic mutations. But these point mutations, or these single nucleotide variants, can be very significant. And when they happen in germline DNA, there can be diseases like sickle cell anemia, which is an enormously kind of protean disease that has all sorts of manifestations that's due to a single point mutation in the germline DNA of patients who have that condition. In cancer, which is the topic that we're interested in, these changes happen in somatic cells, so cells that aren't going to contribute to future generations, but now can acquire a buildup of these somatic changes that lead to cancer. Some of these changes in cancer are very very frequent. So there are some cancer-associated changes that happen over and over from one, across different patients with the same cancer, and even across different patients with different cancers. And so there are two general kinds of mutations, two classes of mutations to think about. And this isn't including things like translocations that are important to patients with myeloma. This paper doesn't deal with that. There are in general two different classes of genes which, when mutated, can promote cancer. And that involves oncogenes and tumor suppressor genes. Now, oncogenes are super cool because these are genes that lead to proteins that have really critical functions in cells that regulate how a cell grows, all of the inner circuitry of a cell. These can be profoundly active, important genes in a cell's circuitry. And this includes genes like KRAS, some of you will have heard of. People with myeloma can have KRAS mutations, NRAS, BRAF. These are all genes that can be mutated in patients with multiple myeloma and other genes. But KRAS mutations also happen in colon cancer. They happen in pancreatic cancer. BRAF mutations can happen in colon cancer. So these proteins all are forms of enzymes, and their function, by and large, is to they act on other proteins. So let's just imagine I'm a protein here, and I can do other things. I can turn on or off other proteins. I have all sorts of important functions as a protein in the cell, but I only have those functions if I exist in a form that contains a phosphate group that is phosphorylated. And so a lot of these oncogenes belong to a group of proteins called kinases. And what a kinase is it takes a protein and it adds a phosphate group. And now proteins that are phosphorylated in very specific ways can dramatically affect the activity of that protein and its function and its effect on many other proteins. And so the Nobel Prize was awarded to a couple of scientists at the University of Washington back in the 1950s because they figured out that protein phosphorylation is a key way of regulating the activity of proteins. And so so in general, bottom line is oncogenes, very active proteins that normally exist under exquisite control by a number of different processes that happen within cells. And when they get mutated, when they get out of control, all hell can break loose in a cell. And so these are proteins that you just treat with, these are, a lot of respect. And this is a small list of proteins that fall in this category. Tumor suppressor genes are also thought of as guardians of the genome. And these are genes like TP53 or the retinoblastoma protein. TP53 is obviously important for myeloma patients because it's encoded by chromosome 17 and P53 deletions. When you have loss of P53, now you have deprived a cell of one of the most important mechanisms it has to kill itself if things are going haywire. And so these two classes of mutations affecting oncogenes and tumor suppressor genes together are profoundly important in driving tumorigenesis across a number of different malignancies. And what this paper aims to do is to come up with a dictionary of all of the genes that are involved in this process. And that's really what they're doing. They're using super sophisticated computational approaches to figure that out. You can imagine for a moment, just think for a moment about what I said about germline versus somatic mutations. Let's imagine that I want to get my tumor sample sequenced. I want to sequence the genome of my tumor sample. Okay, I can go send it off for sequencing. I can get the result back. And let's say that I find, oh, hey, I have a mutation in some important gene, and I know that's present in my tumor. How do I know that that mutation wasn't already present in my germline DNA, and because it was present in the germline, of course, the cells that led to my tumor had that mutation. And so there are mutations where you really need to sequence both the germline and the tumor DNA to understand, oh yes, this mutation truly is only in the tumor, it's not in my normal cells. But there are a number of mutations that have been characterized where we know they don't happen in normal cells. The only way that they can be observed is in tumor. But so it's a little bit of a it can definitely be a question sometimes when you're looking at a mutation in a tumor. Hey, did this really occur only in the tumor, or was it part of my genetic makeup to begin with?
Hey, Tony, if I could briefly interrupt you, we have a couple of patients from some questions. We have a couple of questions from some patients.
Yeah, go ahead, Jane.
Also, are kind of at our 10-minute warning. So,
Yeah, thanks.
We could keep these brief. But we have a question from Dana that says, is the myeloma cancer stem cells which survive treatment, including melphalan, is this the reason or one of the reasons why myeloma patients relapse? If these cells survive, would they acquire additional mutations due to being exposed to high-dose melphalan, thus creating a new lineage of aggressive cells?
Yeah, Dana, it's a brilliant question, of course. I would expect nothing less from you, Dana, but yeah, there is a huge concern, right? So I have, like a lot of people on this call, I have multiple myeloma. You look right now and there's no trace of it if you look with very sensitive testing, but it's almost a sure thing that there's some myeloma cells lurking. This is the limit of detection, and they're lurking below the limit of detection. And so I stay on drugs to kind of try to keep them from coming back. But there's a lot of evidence now to indicate that certain classes of drugs can cause a lot of mutations in cells. And one of those drugs, as you mentioned, Dana, is melphalan. So melphalan belongs to a class of drugs that's called a DNA alkylator. It causes DNA damage. And I know that, for example, there is a lot of concern that exposure to melphalan can cause DNA, significant DNA damage, extensive DNA damage, and if that DNA damage causes the cell to die, perfect. But if there are a small, tiny number of cells that have incurred that damage and yet are able to survive, now when they come back, the concern is, Paul Richardson mentioned this, in a number of forums, and to me as well, that there's a real concern that if and when those cells come back, now they might have an additional added mutation burden that make them especially difficult to eradicate. So that's definitely come up as a concern. What's the other question, Ching?
We have another question that said, I noticed in the article that myeloma mutations share some mutations with other cancers, such as skin cancers, lung cancers, and colon adenocarcinomas. Are these known/common secondary cancers associated with myeloma?
Yeah, so you're exactly right that there are some genes that are mutated in myeloma that are also seen as mutations in other cancer types. But that doesn't mean that there's any connection between myeloma, let's say, and a second cancer. It just means that cancers get built from combinations of mutations, and the combination that triggers a normal cell now to become cancerous has a different path in myeloma than in other cancers. So, you can have mutation 1 in myeloma, 2, 3, 4, and now these cells have acquired a special combination special makes it sound too nice, kind of a nefarious combination of mutations that causes this now to grow and become, cancer. And some of the building blocks in these mutations can be, a colon cancer can take the same gene and independently, now having nothing to do with myeloma, all the plasma cells in that patient are fine, but some of the early cells in the lining of the colon can acquire, a KRAS mutation. And now that can be part of the unique recipe of mutations that leads now that cell to grow up and to be a cancer. So the fact that some of these genes play important roles in these different tissues is important and fascinating and interesting, but it doesn't mean that there's any relationship between one cancer and the other in a patient. And for example, there's no reason to think that a patient with myeloma is more likely to have colon cancer, even though both of those can be associated with KRAS mutations. Okay, I can't hear you, Cheng.
Sorry, Tony, we have a couple of other questions. Rebecca asks, what is the translocation and what is the significance of T1114 specifically? Does it change over time, and what are the factors to consider in treatment for T1114 translocation?
Yeah, so that's a great question. So this paper did not describe translocations, but translocations are another very important class of genetic change that occurs in patients with multiple myeloma. And so, each of the cells in our body has, 2 copies of each chromosome, chromosome 1, all the way to chromosome, 22. I think they get smaller. They're aligned according to their length. I think they get smaller as the numbers go up, and then there's an X and a Y. Somewhere in here, there's a chromosome 11, and there's a chromosome 14. And what can happen then is you can have a chunk of chromosome 11 connected to a chunk of chromosome 14. This is a very common abnormality in patients with multiple myeloma. And what that means is that there's something important about this juncture between these 2 chromosomes that's supporting, that's allowing cells that are destined to be myeloma to grow and have a survival advantage and grow over time. It's probably almost everyone here will know this patients who have myeloma with this specific genetic change tend to be sensitive to a drug called venetoclax that inhibits a molecule that's involved in cell survival called BCL2. And there are reasons why this specific translocation is associated with upregulation of BCL2. They're not totally straightforward, super straightforward reasons, but this translocation is in general associated with increased activity of BCL2. Venetoclax can block that, and that's why there's a lot of interest in using venetoclax in patients who have that translocation. And what else, Chang?
And one final question comes from Vicky. She asked, can gene editing be used to correct somatic mutations?
Yeah, great question, Vicky. So, so here's my concern about that. So let's say, we have and the question is again, can gene editing be used to correct the mutations that lead to cancer, and could that be a way of treating cancer? And so let's say, these cells have a mutation, and I'm just going to put a little, X here. This cell has a mutation. These all become bad boys now, and they're going to grow and cause cancer, and you're in big trouble. But let's say you have a nifty way of doing gene editing, and you can, now we're going to correct this. So we're going to say, okay, we've gotten rid of the mutation here, that cell, don't have to worry about it anymore, that cell, we don't have to worry about this one, this one, this one. The problem that the conceptual issue that I have with the gene editing approach is that the primary way to address this problem is the gene editing has to be perfect. Because if one cell with that mutation escapes, now it and all of its other progeny, that's selection, it grows, it keeps, and so I think gene editing has enormous applications throughout medicine that are super important, but my concern about relying on gene editing to fix mutations is that cancer is hard, and if you don't address every single cancer cell, then the small number of cells that escape your intervention have the potential to grow back and cause problems. So I think we're at the end of our time. I want to thank everyone very much for attending. Please put any comments or any suggestions for improving this. Like I said, this is our very first go. And please let us know if you thought this was worthwhile. And thanks very much, everyone, for joining. It's great to have you. Thank you.